Hello, everyone. I'm Siri Vaith, Executive Director of CFRI, Cystic Fibrosis Research Incorporated, and I welcome you to our fifth question and answer period with Dr. Rick Moss. Today is Wednesday, April 22nd. As always, the date is important because the news and information about COVID-19 changes so rapidly. So if you're watching this recording later on, this might not be most up to date. So again, it is Wednesday, April 22nd. For those of you who are new to CFRI and have found this recording, I do encourage you to go to our website, www.cfri.org, to find out more about our programs. As always, I must note that the information presented in this Q&A is not meant as your personal medical advice or treatment or diagnosis, so please always consult with your care team before making any changes to your health regimen. And so now we are very fortunate to return with Dr. Moss, who has generously shared his time with us for weeks. And it was interesting, I was thinking back, the first time we started this Q&A was at that town hall. And at that time, which was March 17th, I think just slightly over 4,000 people had been diagnosed in the United States with COVID-19. So it's just remarkable every week to see how radically things have changed. We're grateful to have you with us. For those of you who don't know Dr. Moss, he is a preeminent CF clinician and researcher, former director of the CF Center at Stanford and professor emeritus of pediatric medicine at Stanford. So we're honored to have you with us. And we have a lot of questions ahead of us. Welcome. So we will start, as we do every week, with the current report for the US on numbers of diagnosed cases and deaths for COVID-19.
Yeah, every week it seems that it's a vastly different number. Today we have 820,000 cases in the United States, and the death toll is pushing up to 50,000, 46,000 today. By most models, we're really in the apex region of infection in the first wave right now. So hopefully within a week or two, the rate will start to be going down, but we'll have to see. We'll look at the states also, and almost half the cases are situated in the Northeast around the New York metro area, with Massachusetts also now becoming the third most populous state in terms of cases. We're starting to see more of the center of the country and the South with more hotspots as things diffuse out from the major metro areas into more suburban and rural areas.
And I'm jumping ahead probably to other questions, but that does beg the question about spread. Why is it so prevalent in those eastern states, A, and B, with the spread to the growth of cases in the southern states, is there any relation between the sheltering or not sheltering in place policies?
Well, as you know, we have a real mix of attitudes in different states. Most of the governors are really hewing to the experts at the CDC and the NIH in terms of recommendations about maintaining social distancing. There seems to be emerging push in the South, particularly, to go to earlier openings. That's, of course, been urged on by President Trump. And I know that states like Georgia, Tennessee, South Carolina, Florida, Texas, they all have plans for, these are all staged openings. They're not just going to kind of turn the light switch on. But the point is that even in those states, the mayors and municipalities are very concerned that without adequate testing, this is going to lead to new surges of cases. So we'll just have to see what happens.
So somebody has written in, I heard the CFF is now tracking CF and COVID-19 cases in the US, and that last week there were 14 cases. Has the number grown? And what have we seen as far as severity of symptoms for those living with CF compared to the general population?
Right. So the first real official kind of announcement from the CFF was on the 16th, just 6 days ago. So those 14 cases are still the only public information that the foundation has provided to its network of centers and care providers. They do specify that at that time, 328 tests in CF patients had been conducted according to what people are sending to the registry. That's really the only way they can get this information, if people are submitting that data to them. But they did say they'll be periodically updating it. So I don't have anything beyond those 14 cases from the 16th. Europe also has its own tracking system now, and according to a website called CF Europe, on March 31st, 9 sites had reported 16 cases in Europe. Characterization of the clinical courses have not been reported, and all we have really still are some of these social media stories. And the sort of the spectrum of severity has been wide. Some patients with more advanced disease, some with very mild disease, and the fact that everyone is either stable or through it is pretty good news, but it's, at this point, you can only regard it as anecdotal. There's really no conclusions you can draw from that amount of information. It's really very limited, especially the fact that the clinical characteristics have not been described. I do know that, especially in Europe, and I think the foundation is also working on this, there's going to be efforts to get more sort of granular information about the nature of, who these people are, what the level of baseline diseases and how they've fared, and also maybe what they've been treated with. But we're not going to get that information soon, I don't think.
Mutation of the virus. Some of these questions were things I had not known about or thought about. We know that this virus can mutate, and there are reports referencing more than 30 strains of COVID-19. How many strains are we aware of since the initial outbreak of coronavirus? Are the strains vastly different in symptoms, therefore making it harder to detect?
Yeah, so the question about viral strains is somewhat difficult to answer because of the level that you want to get to. Mutations can occur and do occur very frequently, especially with RNA viruses like these coronaviruses. It's generally thought that most mutations don't increase severity, they actually decrease severity. If the virus is already well adapted to its host, which this virus seems to be beautifully adapted to us, it's very easy to get antibodies, and if anything, the mutations will tend to make it milder so that it can get passed on from one person to another. If it's a very lethal mutation and kills everybody it infects, it's not going to be able to spread. So evolution is a funny thing. The adaptation actually would serve to push it in a milder direction in general. Having said that, we know that there's efforts by geneticists to trace viral genomes, and they have found now, the most recent publication was about 30 different strains that have been detected. What we don't know is how that relates to what kind of disease each of these might cause. Initially, it was the Chinese that identified 2 major strains, which have been called L and S, and it's not clear that one is more severe than the other, but we do know that the usefulness of these analyses has been mainly so far in tracking how the virus is spread. This is how they determined, for example, that the New York hotspot really probably came from Europe rather than China, which is interesting, right? And most recently, I think the New York Times today reported that the strain in Washington State, which was the earliest one detected here, showed up on the East Coast very soon afterward. Just indicating how easy it is for these strains to spread around the world. But we still don't know enough about the relationship between these genetic changes in the virus and what it might mean for the level of disease or how easily it's transmitted. And those are important questions. I think the good news is that in general, the lesson from biology has been that most mutations have not made things worse, but generally tends to make things less worse.
So related to that question, somebody asked if there are these strains that they think came from Europe versus the first strains arriving from China, Is there a difference that they can tell yet in severity from source?
Yeah, not yet. I don't think it has reached that level of sophistication at all. Eventually, maybe we'll learn that. There was one study from Iceland which actually showed 7 different strains in Iceland, just in that very small country, which has less than 500,000 people in it. So clearly there's lots of mutating going on. I don't think people should become too concerned about that at this point. Like I say, most of the mutations are not going to be more dangerous. The virus is already wonderfully adapted to getting into human beings and residing there. And the evolutionary pressure will actually be to dampen it down so that it can go on to a new host.
When some people are testing positive again after they had been given the all clear, do we know if it is a reinfection of the same strain, it just they never really were over it all the way, that the viral load was low enough it wasn't detectable and then it flared up, or is it a reinfection? Do they know yet?
I think it's more likely that it's a reactivation of the same virus. There's no firm answer on that at this point. Using some of those genetic analyses may help sort that out. But we do know that the virus is present for longer than we initially thought. There was a very recent study from China, which looked at, besides respiratory samples, they looked at stool and serum. And the median duration was more like 2 to 3 weeks, with some variation between the source, and certainly longer in people that were more severely ill than those with mild illness. So I think it's likely that there may be periods where it's not detectable and then comes back from perhaps another part of the body, or is able to spread again within the body, but we don't know for sure at this point.
Well, it certainly makes the argument about longer periods of quarantine rather than shorter, longer periods of sheltering in place than shorter when you think it can. It seems like 2 weeks has been the operative number, 14 days, but it seems like 21 was.
Yeah, and according to this, according to the most recent Chinese study, in the mild cases, that is people that were not severely sick, not out of the ICU, the median was 14 days, but it could go up to 21 days. And people with severe disease, the median was 21 days, could go up to 30 days. So it is kind of trailing out longer than the earliest reports it would indicate.
Let's see. Well, I'll say personally, last time I was watching the news, they had a story on a teenager who had these sores on his toes, was asymptomatic for other COVID-19 symptoms, but then they tested him and he indeed did test positive. And there are other doctors who have said anecdotally, these rashes have been appearing, and maybe that's yet another thing to follow. And then lo and behold, this morning in the Washington Post, there was an article about blood clotting, which it just seems like the more we know about this, the more complications it seems to have. What can you tell us about this blood clotting that occurs with COVID-19?
Yeah, I mean, this is pretty well known now to the people that work in the ICUs and see the sickest patients. It's actually quite common that they're seeing various kinds of clots, both small clots and also larger clots in the lung that can be called a pulmonary embolus, for example. These seem to be fairly common. So one of the things the virus seems to be able to do is to trigger the coagulation system in the body. And you can actually track that by measuring certain things in the blood. There's something called the D-dimer, which is part of that system. That goes up very high and is a good predictive factor for having predicting bad disease. So yes, the coagulation system does seem to be activated in patients that have that are not doing well. Obviously, not many people get infected and have no problem with that, the majority. But for those at the sick end of the spectrum, yes. And skin rash, that's something that we see in an awful lot of different viral diseases, so it's no surprise there also that various skin rashes, some of those may be also evidence of thrombosis or clots that occur in the skin area, but it may be also other kinds of things that occur with various viral viral illnesses, so I'm not surprised about that either. In general, as time goes on and there are more and more cases, it's becoming clearer that more and more body systems are being involved. The GI system, very frequent diarrhea is one symptom. The central nervous system, many people are having trouble with various neurological problems. If you've been watching Chris Cuomo, you know how he's talked about how it's affected his thinking. This is a very common story now for people now reporting how they've, done with this disease and certain other more serious issues like strokes and so on. Even paralysis has been reported, Guillain-Barré syndrome, which is a form of paralysis. So clearly the virus is affecting many body systems. The kidney is another one, and many patients at the severe end also are needing dialysis because of kidney failure. So yes, this is attacking the whole body. The lungs are the sort of the main focal point, but it's not just a lung disease, it's clearly a body disease.
It just makes me so anxious to have more testing, right? Because there are so many things. I think the general public, the main thing still is sort of the headache, the fever, the cough, and there are all these other symptoms that might be indicators, and people are not going to be tested. So I
think the biggest unknown now is why this tremendous variation going all the way from completely asymptomatic carrier, right, at one end to
you just froze.
Everything in between. Why this vast difference? And that is really a big mystery. Certainly some of the risk factors have been identified: age, diabetes, obesity, hypertension, underlying chronic lung disease, but still tremendous variability in how sick or non-sick people are. And we need to keep remembering, the good news is that for the vast majority of people, this is not a life-threatening illness. The problem is that it is for those high-risk groups in particular, and the occasional person that looked perfectly healthy. You read these reports of the 20 or 30-year-old who was in good health, athletic, so on, and wound up dying of this disease. So we have a lot to learn about what it is that makes the response so variable.
We understand that numerous CF researchers, biotech companies, pharmaceutical companies have been forced to postpone clinical trials due to COVID-19. And as a CF researcher who's been involved in countless clinical trials, what can you share about the impact of these delays, and what happens when there's a 2 or 3 month gap before you can restart? I would imagine you'd lose more than those 3 months.
Absolutely. Just getting restarted again and losing funding, losing timelines, personnel changes, just getting people, getting resourced back into the trials. All these are big problems. And yes, unfortunately, I think CF research has been set back tremendously by the fact that virtually all CF trials have been suspended while people focus on dealing with this. And perhaps in the next phase, as things start to recover from at least this first wave, they'll be able to restart some of these trials. But it is going to make it much more difficult. And just practically speaking, resources within industry, within the biotech industry and pharmaceutical companies, they've also switched resources to try and develop their own treatments and trials. So that's going to take away from other needs. And you see the same effect in clinical care with, canceling of clinic visits, canceling of elective surgeries. Just getting the whole system back on track is going to be a very painful, slow, and expensive process. And it's just another sector of the economy, if you will, that's been hit hard by this pandemic.
So circling back to the question of vaccines, can you report on any further research and development for a vaccine for COVID-19? Will it be, if so, will it be a live vaccination, do you think, or an inactive vaccination? And as a person who is post-transplant, I would hope that it would be inactive.
That's interesting. Well, the vaccine space in this area is amazing. There are at least 86 different vaccines being investigated right now. So it's pretty overwhelming. And they run the gamut from DNA vaccines, RNA vaccines, inactivated vaccines, live attenuated vaccine, what they call non-replicating viral vector vaccines. Probably the biggest single subgroup are what's called protein subunit vaccines, where they take a certain part of the virus and produce that by biotechnology and use that as the agent. So all that work is going forward. There's 3 or 4 now that have entered clinical trials in various countries. And it's going to be, I think, difficult because of the large number of different approaches and the difficulty of doing a vaccine during a pandemic. This has never happened before. So we're going to have a rocky road ahead. But I do think there's reason for optimism. There's one group in England based at Oxford University who are using one of these approaches, who think that they may be able to start larger scale clinical trials as early as September. So they've already developed this protein subunit for SARS, and it may be cross-reactive with COVID, so they can move more quickly. We'll have to see how that pans out. I think eventually there will be a number of vaccine candidates brought in. The question of the person who's immunosuppressed, we don't know if, I think it's doubtful that a live attenuated vaccine will be the one that emerges as the winner. I think more likely some of these other approaches. So hopefully that question won't even arise, that what comes out is going to be safe for everyone. But safety is, of course, the most important thing. There is a yellow flag here, which is that they have already noticed with SARS vaccines that some candidates actually produced enhancement of disease in experimental animals. So we have to be very careful on the safety side moving forward with these approaches. I think we'll have information within a few months about whether that's an issue, because some of these animal experiments are ongoing right now, as well as the early clinical trials. So the vaccine area looks promising. What we don't know is whether some of these genetic variations we discussed earlier may make it necessary to have a changing vaccine. Remember, for influenza, we never got to a single vaccine. It's a seasonal vaccine every year because the virus is changing so much every year. We don't know if that's going to be the case for this yet. Obviously, if that is the case, it complicates things greatly. We also don't know whether the particular antibody responses are going to be fully protective and how long that lasts. There's just a huge amount to learn here. And finally, antibodies are one arm of the immune system that people are focused on, especially with the antibody tests that are now being done. But there's another arm of the immune system called the T cell or cellular response that may actually be just as important. So people have to look at that also. So we have a huge amount to learn. Remember, again, we didn't know this virus even existed 3 months ago. So we just have to be patient and see how the science progresses. The good news is that, the amount of attention being paid is unprecedented in history. So I think these answers are going to come fairly quickly, not as quickly as people would like, but fairly quickly.
Right. And, let us just hope, I have to confess that when this was first starting, before really there were only 1 or 2 known cases in Washington, that I was like, well, think about the flu, over 20, 30,000 people die every year from the flu, and half the people don't even get a vaccine. Like, I was definitely still grappling with what's the difference. Obviously now I know the big difference, and but I do hope moving forward that this, has transformed Americans' public health understanding and the importance of these things, that it's because we'll just never be protected if we continue to have this split. So
yes, and remember that before this thing hit, there was a fairly powerful anti-vaxxer movement in this country. And we've seen outbreaks of things like whooping cough and measles that are due to people refusing to have their kids vaccinated, immunized. And hopefully this will be a gigantic lesson for the country in terms of what vaccines have meant to humanity. People just have very little grasp of how precarious life was before we had vaccines. And as recently, as this decade, we've had problems because of low vaccination rates. And once this vaccine is out there, certainly I think it will be adopted very widely. And there will be issues about who is producing it, who gets it first, how much is available, who's paying for it. These are all issues that will follow the science of the actual production of the vaccine. So we're embarked in a big new era of dealing with this whole public health issue of immunizing, something to which humanity has not been exposed to in the natural world until now.
Let's see. Well, studies from Stanford and USC in California are showing that antibodies are much more prevalent in the general population than we thought. How likely is it then that many of us have already had COVID-19? And is a blood test the only way of knowing if we have antibodies present?
That's the main way that we know these days whether or not you had exposure to the virus, because your body has recognized it and made it an antibody response. Remember that both the Stanford study and the LA studies, and these are the first in the country, I think, that have looked at this and publicized their results, between 3 and 5% of the population shows evidence of having been exposed. So remember, that means that 95 to 98%, 97% of the population has not yet been exposed. Think about that, flip those numbers. So people are focused on the fact that, wow, so many more people have been exposed than there are cases. 50 to 80 times the number of cases have been exposed. But flip that over, 95, 97% of the population, at least in California, has not been exposed. And if you go to other areas of the country with less travel and so on, it's probably going to be below that. So people talk about herd immunity. You need about 50, 60% of the population to have antibodies to have so-called herd immunity. So that's an indication of how far away we are and how vulnerable we are as we move into this opening up phase in various places. Again, 95+% of the population is going to be vulnerable. So without the social distancing, what's going to stop that? The answer is it's not going to be stopped. So if the openings are not done with extreme care in the presence of enough testing, you're going to see new waves that occur as these states go or areas loosen up in terms of their restrictions. And that's the unfortunate reality that we're dealing with here.
I mean, I have to say, with all the protests that have been on the news and seeing the people in Sacramento this week and in Denver over the weekend, and I just thought, well, there's a hot spot. I mean, I immediately see these people clustering, and I think it's mind-boggling to me. There is no way that no one in that group.
But we have to remember this is a media effect. If a couple hundred people show up, to say, "Virus be damned, I'll take my chances," it gets a headline, and people see the video. But if you take a poll, the vast majority of the population says, actually, we don't want things to get loosened up prematurely. We really have to remember that distorting effect of media coverage on some aspects of this. And I think this is a good example of it. So we need to not be misled by those kinds of events.
Mm-hmm. So the next question. We've talked about lots of different cleansers and cleaners that we can use to disinfect things. What about UV light, UVC in parentheses? Does it kill COVID-19, particularly on items like phones and keys?
Well, I'm certainly not an expert in this, but the question did send me scrambling to the internet, and I realized that there are 3 types of UV lights that people talk about, A, B, and C. And UVC is the one that has been shown to be effective against SARS. I don't know of publications yet looking at particularly COVID, but its close relative SARS is killed by UVC light. The problem with UVC light is that it's very toxic to the skin. So you can get sunburn very, very easily, much higher rate of skin cancer with more exposure. There's something called far UVC, which is some kind of tweak on how you generate UVC light. I don't think these devices that create far UVC are generally available. So it's not a method that most people have recommended for disinfecting. And we have to go back and remember this is a virus that takes part of the cell membrane with it, creates a lipid or fatty kind of envelope around it, which is very susceptible to disruption by soap, detergents, and alcohol. So those things can be used instead.
If we, and I think this is the final question for this week, if we don't have a vaccine, but we are fortunate enough to have effective therapies developed and approved, would you still recommend that those with CF shelter in place until there is a vaccine?
Well, we need to learn more about the effect of this infection on people with CF. One of the interesting things that's emerged in the last couple of weeks, there have been some studies suggesting now that people with asthma are really not at risk like people with COPD are. So there may be actually nuances within chronic respiratory disease between different particular disease states and susceptibility to having a severe episode of this infection. So we need more information on that. And I hope that comes as more registry type data is provided to CFF and also over in Europe, the European CF Society. But in general, I would say that if there's an effective treatment, we need to know, first of all, what's the target, or is it people who are being prophylaxed, taking something before they're infected? Is it people that have been diagnosed but are not sick, the asymptomatic? Is it people with mild disease, or is it people that are hospitalized with more severe disease? Different treatments will go into those different niches in terms of treatment. So if there is an effective treatment, the one question is, effective for what particular phase of this problem? In general, I think if there is some effective treatment, it'll make it much easier to say you can go out if you take certain precautions. And people with CF continue, as they have before, to use those precautions for cross-infection purposes, especially those that might have had transplants, obviously. So in general, I think the answer to the question really depends on what kind of treatment we're talking about, for what stage of the disease, how effective is it, what are the dangers, and you're going to have to make some judgments based on that kind of information, which we don't have yet. The important thing to note, as of today, there is no effective specific treatment. And there's more and more studies that are showing problems with hydroxychloroquine, azithromycin, some of the antivirals. The two things that at this point to me still look very promising are remdesivir for severely affected patients and convalescent plasma for severely affected patients. So again, those two things really apply to people already in ICU or at least hospital settings rather than, before that. So we have to wait and see if we can get better treatment.
Once again, Dr. Moss, thank you so much for sharing your time and your expertise. You've just been such a fantastic voice for us, a person to go to really to address our specific CF questions, and we are all grateful.
Thank you. Stay safe.